Okay. This is actually the start of a new chapter. Again, guys, warn me if I go off screen up there. You have to be the eyes in the back of my head. All right, so new chapter, from DNA to protein. Now, admit we're skipping over just a little bit. We're a little behind, got to do a little catch up. So in a normal year, it'd be a day or two we'd spend on some really cool experiments that were done in the early 1900s, 1910s, 1920s, 1930s, 1940s. And they're actually pretty cool, and the results are really interesting. But no huge time for it today. So if that's something you think you'd be interested in, let me know. I can get you plenty of reading on it if you're interested. But we're just going to kind of skip ahead and get right to the meat of our next unit, from DNA to protein. So starting off, so here's kind of the idea. We all know what the genetic material is. It's no surprise to us. We've grown up with it. You guys have heard about it for years. You even may kind of know how it works already. You probably covered something about it back in 6th or 7th grade. But that hasn't always been the case. I was saying those experiments back in the early 1900s, we're talking about 100 years ago. It's not that long ago. About 100 years ago, we had no idea what the genetic material was. I want to take that back. We had an idea. We just weren't sure, and we certainly didn't know how it worked. So the genetic material for us, again, is kind of common knowledge, but again, 100 years ago, it certainly wasn't. But we did know it had to be able to do two things. Number one, has to be able to replicate itself. How do we know that? Well, because you are 37 trillion cells. And each of those 37 trillion cells has the exact same DNA, the exact same genetic material. So there's proof right there. That whatever the genetic material is, has to be able to replicate itself. And then the second thing, has to be able to serve as the instructions for life. So those are the two things that DNA has to be able to do. And that's actually what this whole chapter is going to be about. We're going to focus on how it does those two items. 9.1, first unit. The structure of DNA. All right, this is where we're going to start getting a little artistic. Guys, it's nothing more than simple shapes. And put them in different combinations. Slow down and concentrate. Sometimes maybe use your eraser. It happens. Slow down, concentrate. That's kind of the point of taking notes, right? As you're slowing down, you're thinking about what you're doing. Hand, eye, thought process. So that's why we take notes. So slow it down. These are your simple shapes. Kind of space it out. I'll do my best to help you guys along. I'm going to skip down a couple lines here so I can, write some more stuff in here later. I'm going to start. Circle. I'm going to connect that circle to a pentagon. Yes, we do kind of have like this elbow right here, and yes, that elbow is kind of important. This pentagon, I'm going to connect to another pentagon. And I'm going to connect that pentagon to a hexagon. Like I said, artistic, right? Okay, we're going to add, now try to keep it in line with this circle. We're going to add another circle. Try to keep it in line with that circle and connect it down here, like that. Draw another elbow, and again, we're kind of repeating what we did up here. Connect it to pentagon. This pentagon is going to connect to a hexagon, and that's it, just the hexagon right there. And let's do it again. This time I'm going to connect it to two rings, but I'm going to start with my hexagon. Is that terribly important? No, not really. So I got my two rings there. One's a hexagon, one's a pentagon. And is that terribly, terribly important? No, not really. But details matter, and that is one of those finer details. And we'll do one more. Boy, my sizes got kind of distorted there. Theoretically, they should all be the same size. I'm just terrible at art. Oops, ran off the page there, didn't I? Come on, guys. Gotta be the eyes in the back of my head. This time we're connected to just one, and it's going to be a hexagon. I'd love to be able to tell you we're done, but if I did, I'd be lying. To continue, for me at least, it's going to be a little bit easier to flip my page upside down. Pretty much. Someone asked, are we doing the same thing? Pretty much. Want to make sure that they line up. So I'm going to take my time here, and I want my shapes right in there. So my circle kind of up above there. So there's my circle. Draw the elbow. There's my pentagon. Now, because this one here was only one ring, see these were two rings, this one's only one ring, I'm gonna make sure that this one has two rings. And I'm actually gonna start with a pentagon, and then hexagon. Okay, this next one then has two rings on this side, so guess what? I'm going to put one ring over here. Hexagon. We'll do it again. Don't forget your backwards elbow there. It is actually kind of important, that backwards elbow. Talk about him more in a minute. But first of all, it's kind of keeping us in line. If you don't have them, your whole thing starts to tilt. All right, this one here only has one shape, so this time we're going to put two rings. I will start with my hexagon this time. All right, we're almost there. One more. On this side, again, we have a two-ring structure, so this time we'll just put a single ring. Hexagon. All right. How about that? That's pretty artistic, right? You're well on your way to being biologists, all these pentagons and hexagons and circles. Good work. Let's take some notes now, huh? We'll be back to this page. We'll come back. So those of you guys who aren't quite there need to see something, we'll be back in a few minutes here. Let's see which way we go for the next page. Yep, this way. Cool, cool, cool. Starting off with the nucleotide. If that word sounds familiar, it should. We actually have talked about these quite a bit. We talked about them first semester when we did our macromolecules. There's 4 different macromolecules: proteins, lipids, carbohydrates, and nucleic acids. Guess what the NA in DNA stands for? What's that? Not the D. Drop the D for now. We'll cover that in just a second. Just the NA part? Yep. That NA stands for nucleic acids. So the nucleotide is the building block of nucleic acids. So DNA is made up of the repeating monomer unit called the nucleotide. It's kind of like a train car. I don't know if you guys have ever been stopped on the road and like a train had to go by and you just kind of had to sit there and wait. You guys ever count the cars? See how long your trains are? Got to 140 one time. That's like the longest one I can remember. It's a long train. I was there for a while. But anyway, those train cars are kind of like the monomers. The whole train is like the macromolecule. It's this big, long molecule, like a big, long train, and it's made up of these repeating subunits, like train cars. Yash, I appreciate asking questions. Sit tight. We'll get there. We'll get there. You're ahead of us. Hang on. All right, so flipping back. Drawing side, I'm going to put a box around just one nucleotide. So here is one nucleotide. And you can see in your drawing there how we have several nucleotides all linked together, again, like train cars, making this big, long chain. Nucleotides have three parts. One, or A, or whatever, first part. A pentose, which means 5 carbon sugar. Good, flipping back. We did something similar when we did photosynthesis. And we always said the corners of the hexagon in photosynthesis were all carbons. Well, yeah, that's actually kind of the same story here. So this pentagon, this is our pentose sugar. So that's our sugar there. Now, it doesn't seem like it's important, and today you will not see the importance, but it is very important that you see where and how we number these. So this first corner right here, that's what we call the 1' carbon. It's that little like apostrophe after the 1 that's called the prime symbol. So we call that the 1' carbon. Then we go 2'. This one right here is 3'. Up here in the corner by the S is the 4'. And then you might think that we go to the top of the pentagon for 5, and we do not. Don't get ahead of us. We do not. The top of the pentagon is actually an oxygen. It's actually really cool how the chain kind of wraps around itself and makes that shape. And it's an oxygen up top here that holds it all together. So it's really kind of cool, but we're not going to talk about it this year. You can talk about it more next year in chemistry. This 5th carbon is over here. It's the elbow that we were so certain to draw every time. That's our 5th carbon. And again, it is really important that we see these numbers. You won't see it today. Over the next couple of days, you should start to see why those numbers are so important. I'm going to do the same thing, not on every nucleotide, but I am going to do it on the one down here at the bottom of the other chain. So flip over my paper. Again, here's the 1'. It's attached to the structure in the middle. 2', 3', 4', and again, 5' is kind of that elbow right there. So that's the first part of all nucleotides, a pentose sugar, a 5 carbon sugar. We also always have Ooh. Phosphate group. Hey, again, this should look familiar, a phosphate group. We have worked with these as well, back to our chapter in photosynthesis. We worked with phosphates. Remember, ATP was adenosine triphosphate, which were 3 phosphates. Well, same thing. That circle is going to be the phosphate. And the third part, a nitrogenous base. Nitrogenous means it's nitrogen containing. It contains nitrogen. I don't know if you guys have for Latin anymore. Do you guys take Latin? The suffix ous, os. Not just in this word, but in most English words, ous usually means full of or containing. Has. So it has nitrogen. And so this structure here in the middle, or here, or down here, these are the nitrogenous base. One thing to add about that 5-carbon sugar, sorry, got to put this in just a minute ago. In DNA, this makes sense in just a second here. In DNA, that 5-carbon sugar, that pentose sugar, is called deoxyribose. Mm. 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 That helps us kind of see where the name DNA is coming from. Deoxyribonucleic acid. All right, back to our nitrogenous bases because we're not done with them yet. They're actually quite important. The phosphates and the sugars, they don't change. Nothing unique about them. But you already kind of noticed those nitrogenous bases change. Some are long, some are short. So those guys are kind of our fun guys there. There are 4 different bases. You probably already know them, at least by their 1-letter abbreviation. Adenine, which we just call A. Guanine, called G. Thymine, which we call T. And cytosine, which we call C. So there are 4 bases. Again, we usually just refer to them as the 1-letter abbreviation. I do think you guys should know the name and be aware of it because you'll see it and hear it enough. But again, generally just call them by the 1-letter abbreviation, A, T, G, and C. So in addition to the fact that we've got 4 different bases, there's also 2 different types of bases. Hey, let's zoom in and help. Did zooming in help? Looked like it wouldn't. Good. Excellent. All right, so we've already kind of drawn the 2 different types of bases. You guys have already kind of seen that. The first type we call purines. Purines have 2 rings. So again, we're drawing our structures on the other page. We made some of them with 2 rings, a little bit longer, and the 2 that are purines are A and G. And then the other type are called pyrimidines. And they just have 1 ring and they're much shorter. So purines and pyrimidines. A and G are purines, they're the big ones. T and C, thymine and cytosine, they are the shorter ones. They're the pyrimidines. All right, so here's our 2-ring structure we drew. Guess what? That one's a purine. Here's our 1-ring structure. That one's a pyrimidine. The one up top, purine, this one specifically is G. How do I know it's G? Well, because G has the pentagon closest to the sugar. And then out here in the middle is the hexagon. Is that important for this class? Not at all. Not at all. Again, like I just said, it's just the details that matter, and that's just one of those finer details. But certainly don't go home and memorize that. That's not something to study. Down here then, pyrimidine, we're going to call that one T. Our next purine, we're going to call that one an A. And again, not something worth studying at all, at least not for this class. But the difference between G and A is G has the pentagon close to the sugar, whereas A has the hexagon closest to the sugar. Not important, just again, one of those finer details. And then finally down here at the bottom, we're going to call this one C. So again, those nucleotides are the monomer units. They're like the train cars. The train, they're coupled together like train cars. They're linked together by what we call a phosphodiester bond. And that occurs between the phosphate of one nucleotide and the sugar of another. Let's flip back over here. Right here is a phosphodiester bond. We have a phosphate of one nucleotide, the sugar of another. Right here is a phosphodiester bond between the phosphate of this nucleotide and the sugar of that nucleotide. And right here is a phosphodiester bond. So these phosphodiester bonds between phosphate and sugar, phosphate and sugar, phosphate and sugar, they create what is called the phosphate sugar backbone of the DNA molecule. So it's the backbone of the DNA molecule. We refer to that a lot, actually, when we talk about DNA. Got these phosphates linked to sugars, linked to phosphates, linked to sugars, linked to phosphates. right, so another box around our sugars and phosphates. Actually, I'm going to draw it on this side too. And these are what we call the backbones of the DNA molecule. We're almost there. Getting closer. Filling up this page more and more, huh? Keep in mind as you guys are making all this, if you are out of room and you want to go to the next page, that's fine. Or when I go to the next page, if you want to continue because you still have room, all of that is perfectly all right. Again, these are your notes. So if they look different than mine a little bit, that's fine. No biggie. So now that we've got these long chains of nucleotides, these long chains of monomers linked together, now we're going to get a bigger picture of the molecule of DNA. So the overall molecule of DNA then is what we call double-stranded, meaning there's two strands, two backbones, and they run in what we call opposite directions. It's known as anti-parallel. So you kind of see the idea of anti-parallel there. Two lines, but they're moving in opposite directions, parallel to one another, but in opposite directions. Now you guys don't necessarily have to flip back. We're not adding anything, but starting to point out that anti-parallelism. This pentagon here, we numbered it 1, 2, 3, 4, 5. Look, 5 is at the top, so it's moving that direction. And the one at the bottom is opposite of that. We've got 5 at the bottom, which technically means 3 is at the top. If we were to number this one here, 3 prime would be at the top. So in one end we have 5 prime at the top, the other end we have 3 prime. So they're anti-parallel to one another. They're running in opposite directions. What holds those 2 anti-parallel strands together is in the middle, they're nitrogenous bases, the A's, T's, C's, and G's. They pair up and hydrogen bond together. So it's phosphodiester bonds holding each nucleotide in line, and then one strand is connected to the other strand with hydrogen bonds. Those nitrogenous bases in the middle bond together. All right, I'm ready to flip a page. Everybody else ready? We need another couple seconds here. I'm kind of indented here because I'm really underneath where I just was.
Can you go back to the last page for a quick second?
How you guys holding out hunger-wise? Is it all right, or getting to the point where you're wishing it was lunch? And not wishing it was lunch just so you could get out of here. Yeah, okay, we all have that. But like, how many of you guys are starving right now? Normal, we don't do eating, but next week, bring a snack. I guess I'll allow you to figure out some way, allow you to munch on a snack or something if your stomach's absolutely killing you. I agree. It's kind of a long period. You people at home too, if you're coming in on Thursday, if you know, we got sea lunch, it's a late lunch. If you remember, you can bring in a snack. We'll figure out some way to get you guys to eat it. Chance to eat it. Are we good on this page? Cool. I'm flipping it. So I'm kind of indented here because, well, this is a continuation of where we just were. So if you are still on the same page or can continue to write, go for it. But I'm really just like underneath the part that says the 2 strands connect in the middle when the nitrogenous bases hydrogen bond with one another. And the idea here is that a purine always pairs with a pyrimidine to keep the width of the ladder constant. And think of those like two rungs coming together in the middle, like rungs of a ladder. And you always want a short base, a pyrimidine, to pair with a long base, a purine, to make sure that you have a consistent width of your ladder all the way down. You don't want two long bases to pair together and you go really, really wide, and then two small bases, two pyrimidines coming in, you get really, really short so they can match up. You have this like really curvy looking DNA strand, would not be very stable, would not be very good, just wouldn't work. So we always make sure that a purine is paired with a pyrimidine. That brings us to Chargaff. I believe he's a Frenchman, Chargaff. I'm not 100% sure if I spelled his name right. I especially think there might be only one F there, but c'est la vie. And Chargaff was one of those experimenters that we talked about at the beginning of the lesson, or I believe it was in the 1930s, again, before we knew that DNA was the hereditary material, before we knew what DNA did and how it worked, Chargaff provided a huge piece of the puzzle. Chargaff did chemical analysis on a lot of different DNA strands, a lot of different organisms, a lot of different DNA strands. And what he found, he thought this was really weird. Now, he didn't explain it. But what he found when he broke down the chemicals and he looked at what was there, is that the number of A's always equaled the number of T's. Didn't matter if you looked at human DNA or frog DNA or tree DNA or bacterial DNA. It didn't matter. The number of A's always equaled the number of T's. And the number of G's always equaled the number of C's. Which is, again, a really, really kind of weird thing that he found out. He couldn't explain it. He didn't explain it. He just figured out that it's true. It's a law. A's always equal T's, C's always equal G's. It wasn't until about 10 years later, 10, 15 years later, after Chargaff found this law, found this to always be true, that another group of scientists that included Francis and Crick and Franklin and Wilkins, and they were able to piece it together, and they were able to say the reason why. The reason for Chargaff's law, Chargaff's rules, that A always pairs with T and C always pairs with G. So some things you don't have to memorize. Which base goes pentagon then hexagon? Which base goes hexagon then pentagon? That, not important. But this, very, very important. Yes, you do have to memorize if you haven't already. And again, this might have been something you've worked on, since like third or fourth grade. I don't know. But A always goes with T. Those are straight letters, right? Think about the letter A, you make it with 3 straight lines. Letter T, you make it with 2 straight lines. So the straight lined letters go together, and the curved lined letters go together. You make C with the curved line, you make G with the curved line. So that is something to make sure you memorize. It's something that will come up over and over again. A always pairs with T, and C always pairs with G. Kind of an addition to the note we made up here about the hydrogen bonds. A and T share 2 hydrogen bonds. And C and G have 3 hydrogen bonds. Is that terribly, terribly important? No, but it's a little important. We've actually found that thermophiles, those are bacteria that live in like really, really extremely hot conditions, like hot springs, they actually have more C's and G's in their DNA sequence. And we believe it to be because with 3 hydrogen bonds, they're going to be able to hold their strands together a little bit tighter. In a really hot in the area, you don't want your DNA to come apart. It's more likely to come apart in a super, super hot area. So a lot of those organisms that live in those super hot areas have more C's and G's. 3 bonds is stronger than 2 bonds. They can kind of keep their DNA held together a little bit tighter. So just kind of an interesting fact there. We can make applications of it. Later. Now, just kind of, like I said, an interesting fact. All right, last thing I think we're adding to this diagram. "28 more minutes. We're almost there. Almost there. All right, so first of all, let's go and flip this over. We can fill this chart in now, right? C always pairs with G. A on this side always pairs with T. T over here always pairs with A. And G on the right must pair with C on the left. All right, the next thing we're going to do then is, again, C and G, they're held together with 3 hydrogen bonds. So 3 hydrogen bonds holding together my C and my G. A and T down here, they're only held together with 2 hydrogen bonds. Here's A and T again. So again, we'll put just 2 hydrogen bonds. And C and G on the bottom, 3 hydrogen bonds. Since that's just representing the bonds holding our 2 strands together. All right. Cool. So our 2 anti-parallel strands then twist, creating what we call a double helix. So here's my old model of DNA. So we've got one backbone here, it's in yellow and red, and we've got the second backbone here in yellow and red. So two backbones, and then the rungs in the middle, again, they're kind of like rungs of a ladder. These are those nitrogenous bases, and they're paired up in the middle. They're hydrogen bonds, I guess, represented by the parts in the middle here. And what happens with our strand is it twists. Makes a helix. Probably one of the most recognizable molecules then in the world. I think we've all probably seen some sort of double helix. We use it all the time in biology. It's a really famous symbol anymore. Equating that twist, kind of wraps around. That's our double helix. It's our molecule of DNA. Slow this down just a little bit. Take your time. Artistic effects. Try to make sure they're the same width. And slow it down. Don't be afraid to use your eraser. I do all the time. It's about 10.5 base pairs per twist. So every time you complete a circle, twist around the DNA, you're going to count 10.5 pairs of A's and T's, C's and G's. Well, this is a cool artistic effect, I think. Kind of have it trail off into the distance, get smaller and smaller and smaller. It's just like a little line here. And then we're going to have it wrap around these circles. And these circles are called histones. So here's our histones. These individual little circles. Histones are proteins that kind of help to organize the DNA. Organize and protect it. Histones are found in groups of 8. I know I've only got 4 circles drawn, but that's because there's 4 behind it also. I just don't have the artistic ability to draw 4 circles behind these 4. So you gotta kind of use your imagination, or those of you with more art history than I can maybe try to add in 4 more circles behind these. So it's a group of 8, and that's called a nucleosome, which again is just kind of a more organized way. We're organizing this DNA. DNA, just these histones and nucleosomes kind of twist and turn and coil up and make supercoils. And that's where we get a chromosome. So here's our chromosome. And again, it's a ton of DNA. "15 minutes, almost there. Almost there." All right, just one more level of organization. This will be quick. This is definitely not this whole page. Like I said, just one more level of organization. Nah. Here's one chromosome. And I like to emphasize this. A lot of times I think people associate one chromosome with one gene. This one little region right here is one gene. So a human chromosome had between 50 million and 300 million individual nucleotides, A's, T's, C's, and G's. 300 million, as many as 300 million in one chromosome. That's a pretty large range. 50 million to 300 million is true. It's a very large range. And the reason is because our chromosomes are all different lengths. We have some that are really long and some that are super short. In fact, some of us in here have one that's even shorter than the rest of us have. Talk about that later. Something for the future. So huge, gigantic stretches, really, really big sections of nucleotide is a chromosome. A gene is just one section on that chromosome. It's not definitely not the whole chromosome. Every time you want to make a protein, you don't want to have to read through 50 million A's, T's, C's, and G's. Take forever. So a gene is a stretch, it's just one small segment. And a gene codes for one protein. A gene, at least for humans, is usually only a few thousand nucleotides long. So definitely not a whole chromosome. It's the equivalent of, giving you a book and asking you to read, I don't even know if I could say one chapter, maybe one section of one chapter. Just read 9.1. You have to read the whole chapter 9. Definitely don't have to read the whole book. One particular protein is one small section of the whole chromosome. Last note. Nope. The average human chromosome, so your average stretch of DNA with its gigantically long, coiled, wrapped around histones, nucleosomes, double helix, the average stretch of DNA for humans, chromosome, contains about 800 different genes. So, and one of the things when we get to genetics in a couple chapters, one of the things drives me nuts is when people think, hey, one chromosome, one gene. That's not the case. You get roughly 800 genes on each of your chromosomes. So it's a lot of different proteins each particular chromosome can code for. Still not us. Sorry. Whoops.